Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Biochemical Tests of Bacillus cereus. Table of Contents. Introduction. Morphological Characteristics. Basic Characteristics. Fermentation Profile. Enzymatic Reactions. Key Biochemical Tests. Conclusion. Introduction. Bacillus cereus is a gram-positive, rod-shaped bacterium that often appears in pairs or chains under the microscope. It is spore-forming, producing highly resistant endospores that can survive harsh environmental conditions, including heat and disinfectants. This bacterium is facultatively aerobic, meaning it can grow in both the presence and absence of oxygen. It is widely distributed in soil, dust, vegetation, and various environmental surfaces, especially in agricultural and food production settings. B. cereus is known to contaminate raw and cooked food, particularly rice, pasta, dairy products, and meats, leading to foodborne outbreaks. It produces toxins responsible for two types of gastrointestinal illnesses: the emetic, vomiting type, caused by cereulide toxin, and the diarrheal type, due to enterotoxins. Beyond food poisoning, Bacillus cereus can also cause serious opportunistic infections, including endophthalmitis, wound infections, bacteremia, and meningitis, especially in immunocompromised individuals. Its ability to form biofilms on medical equipment and surfaces increases its clinical significance in hospital settings. Biochemical testing is a crucial step in identifying Bacillus cereus and distinguishing it from closely related species such as B. subtilis, B. thuringiensis, and B. anthracis. These tests help microbiologists confirm its identity based on specific enzymatic activities, sugar fermentation profiles, and substrate utilization patterns, contributing to accurate diagnosis and contamination control. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Morphological Characteristics. Gram stain. Bacillus cereus appears as gram-positive rods under the microscope. In older cultures, or under stress, some cells may appear gram-variable due to cell wall changes. Cell shape and arrangement. Cells are straight rods with squared-off ends, often arranged in pairs or short chains. Average cell size ranges from 1.0 to 1.2 micrometers in width and 3.0 to 5.0 micrometers in length. Spore formation. Forms oval, centrally to subterminal located spores that do not swell the cell. Spores are highly resistant to environmental stress, contributing to survival in adverse conditions and food products. Motility. Bacillus cereus is motile due to the presence of paratrichous flagella distributed all around the cell surface. This trait aids in colonization and movement across surfaces, including food and tissue environments. Hemolysis. Shows strong beta hemolysis on 5% sheep blood agar, forming clear zones around colonies due to complete lysis of red blood cells. This is a distinctive feature that helps differentiate it from non-hemolytic bacillus species like Bacillus anthracis. Colony morphology. Colonies are usually large, flat, rough, opaque, and grayish-white in appearance. often display irregular edges and may exhibit swarming on nutrient agar due to motility. Basic characteristics of Bacillus cereus. Bacillus cereus is a gram-positive, rod-shaped, spore-forming, and motile bacterium known for its role in foodborne illnesses and opportunistic infections. It displays several distinguishing biochemical features, including positive results for Catalase, citrate utilization, and Voges Proskauer (VP) test, along with strong beta hemolysis on blood agar. While it is generally indole and oxidase negative, it can grow in potassium cyanide KCN medium and produce resilient spores. Fermentation profile of Bacillus cereus. The fermentation profile of B. cereus reveals its ability to utilize sugars such as glucose, glycerol, maltose, fructose, and trehalose, whereas it typically does not ferment lactose, mannitol, or xylose. Variability is observed in the fermentation of cellobiose, salicin, and sucrose. Enzymatic reactions of Bacillus cereus. Enzymatically, B. cereus is positive for lecithinase, casein hydrolysis, esculin hydrolysis, and tyrosine hydrolysis, supporting its identification in both clinical and food microbiology. However, reactions like arginine dehydrolase and acetate utilization may vary between strains. These combined morphological, biochemical, and enzymatic traits are crucial for the accurate identification, differentiation, and understanding of its pathogenic potential in laboratory diagnostics and food safety assessments. Key biochemical tests of Bacillus cereus. Catalase test, positive. Oxidase test, negative. Indole test, negative. Methyl red MR test, positive. Voges Proskauer VP test, positive. Citrate utilization, positive. Urease test, variable. Nitrate reduction, positive. Gelatin hydrolysis, positive. Starch hydrolysis, positive. Lecithinase production, positive, used for identification. Casein hydrolysis, positive. Glucose fermentation, positive. Acid, no gas. Mannitol fermentation, negative. Sucrose fermentation, variable. Lactose fermentation, negative. Xylose fermentation, positive. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Conclusion. Biochemical tests play a crucial role in the accurate identification of Bacillus cereus from clinical, environmental, and food samples. The organism is typically identified by a positive Catalase test, positive lecithinase activity, and a positive Voges Proskauer VP test. It also shows strong hydrolytic activity on substrates like starch, gelatin, and casein, which aids in its differentiation from other Bacillus species. The distinctive beta hemolysis on blood agar further supports its identification in lab diagnostics. Understanding the biochemical profile of Bacillus cereus is essential not only for diagnosing infections, but also for monitoring food safety and preventing outbreaks. Correct interpretation of test results allows timely action in both public health and clinical microbiology settings. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!